All right, good evening, everybody. We're going to take I hope 10 minutes to just give you a quick background information on some absolutely amazing experiments that were done that led to the discovery both of what DNA does and the shape of DNA and how DNA works. And so I think that to really understand some of the stuff that you're learning now, you need to understand how they figured out some of this. So we'll start out really quickly. Give me a second to get there. Back to the 20th century. By the middle of the 20th century, genetics was well established as a field of study. It was known that traits were inherited, but it was not known how that process happened. And we're still figuring that out today. Blah, blah, blah. The discovery of DNA's role in inheritance is arguably the most significant contribution to understanding how life works. And there's a whole bunch of experiments that added together to make that. So let me back up and show you this one right here. Before 1940 or so, most biologists thought that protein was probably the molecule responsible for inheritance. Well, you and I know we've studied proteins like 8 billion times in this class. So it's certainly understandable that people would think that proteins were, in fact, the be all end all. They didn't understand that DNA was coding to make those proteins. So let's see how they figured all that out. So the first experiment that happens is through this fine fellow named Frederick Griffith. I'm going to jump ahead and show you. This is a photograph of two different types of bacteria. The bacteria on the left, these tiny little circles, those are called the R Streptococcus pneumoniae, and they are harmless. That's how I remember it. The letter R, harmless. So those are the harmless bacteria. The bacteria on the right, which are these big blobby ones, those are pathogenic types of bacteria. So let's take a look at this brilliant experiment that Griffith did that was fascinating and very difficult for people to understand, even today, for you to understand. But it was difficult for them to understand what actually happened. So he did this experiment where he took two control groups. So he had some living S cells. These are the deadly, the pathogenic cells. And he injected them into mice, poor mice, and they died. Okay, so then he had the living R cells, the harmless cells, and he injected, and they're pneumonia also, but the mice stayed healthy. Then he heat killed the S cells. So he heated them up and killed them, injected them into the mice, and they were healthy. Okay, so the bacteria is dead. It worked. But this is the part that's really crazy. I got to move my picture so you can see it. This last stage. He mixed the dead S cells with some living R cells. He mixed the dead S cells with some living R cells, injected it into the mice, and the mice died. What? The pathogenic ones were dead, and the harmless ones were there, and now the mice died. What's going on? Well, if you can see down at the bottom image there, what's happening is a process that's called transformation, and they didn't know anything about it at the time. But bacteria have the ability to suck up the DNA of dead bacteria that are in the environment. So it would be like if somebody died on the road and I walked over and went and sucked up their DNA, and suddenly I had the characteristics of that person. Maybe my height would change and my gender or sex would change and my skin color would change. That's what these bacteria are able to do, is suck up the DNA of a dead bacteria, and then they become like that dead bacteria. So that's what happened to the harmless bacteria. They sucked up the DNA of the dead bacteria, and then suddenly they became pathogenic. They didn't understand this. The scientists were mystified by what happened, but just be aware that that's what was going on. So then Avery, McCarty, and McLeod, they refined this Griffith experiment. So first of all, they exposed the harmless bacteria to two things, to the S bacteria's protein and to the S bacteria's DNA. Because remember, we're trying to figure out here what's causing the transformation. What's the real inheritance here? Is it the DNA or is it the protein? So they exposed it to protein and they exposed it to DNA. Once only the bacteria exposed to the S strain's DNA were transformed into dangerous bacteria. So if they were exposed to the proteins, they didn't become dangerous. But if they were exposed to the DNA, they did become dangerous. Still not enough evidence. We got to build on that. Okay, so then Hershey and Chase do this crazy experiment, and this is an experiment that is so the kind of experiment that the College Board is going to ask you about. So I just thought I'd make sure we understood this. So they worked with something that are called bacteriophages. So bacteriophages are viruses, and they're wickedly cool. They are viruses that attack bacteria. That's why they're called bacteriophages. Did you know that bacteria can get sick too? The problem with bacteriophages is that you and I, when we get a virus, we might cough and whatever. We have an immune system that fights it off. Bacteria, when they get infected, they die. Like there is no surviving it. So down in the corner here, let's see, give me a second to get there. Down in the corner, this is an image. This is an electron microscope image of bacteriophages. So the green things that look like little robots invading a planet, those robots are viruses. And you can see them injecting green DNA. It's been colorized, but they're injecting DNA into what looks like a planet, and that is a bacteria. And so those are bacteriophages that are infecting an E. coli bacteria. So this is what we did to kind of finally figure out, is it DNA or is it protein that's really the source of heredity? And it's an absolutely brilliant experiment. So the first thing they did, they have this batch number 1 of bacteria and phages, bacteriophages that are growing together in a radioactive solution, and the solution has radioactive sulfur. Sulfur is not ever found in DNA, but it is found in protein. It's found in 2 different amino acids, methionine and cysteine, I believe, are the 2 that have sulfur. So while the bacteria and the phages, the bacteriophages, are growing in this batch of radioactive sulfur, they're incorporating radioactivity into their proteins, but not into their DNA, because the DNA doesn't have sulfur. So anything that's protein, anything in this experiment that has to do with protein is going to be radioactive. So they allow the bacteriophages to infect the bacteria, and then they place the whole concoction in a blender. I am not kidding, literally a blender. They smoosh it all up, which causes everything to fall apart. The bacteriophages get smooshed, the bacteria get smooshed, everything falls apart. And then they put it in a centrifuge and they spin it. Well, it turns out that the bacteria, the cell, its contents, those are heavy. And the leftover stuff from the phages, those are really light. And so the heavy stuff from the bacteria sinks to the bottom, and the phages and their body parts are left over at the top. And then they tested to see where the radioactivity was. And all of the radioactivity was in the top, and there was no radioactivity in the bottom. So whatever had been passed on to the bacteria, it was not in the protein. No protein had been pushed into the bacteria, because the pellet at the bottom that had the bacteria didn't have any radioactivity. The radioactivity stayed floating at the top of the test tube. So bear with me for a second. So then they did the same exact experiment, only they switched solutions. And this time they put them in a radioactive phosphorus solution. This time, that's what affects DNA. Remember, DNA, the backbone of DNA goes sugar, phosphate, sugar, phosphate, sugar, phosphate. So that phosphate has now radioactive phosphorus in it. So they allow the phages to infect the bacteria. They blend it all up, they centrifuge it, and they look to see where the radioactivity is. The radioactivity now is inside the bacteria, because the phages didn't inject a protein into the bacteria. They injected DNA into the bacteria. And so it was a way of showing that the hereditary information of those phages, that what they were transferring into the bacteria wasn't protein. They were made of protein, but it wasn't what they were injecting into the bacteria. What they were injecting into the bacteria was their DNA. And in this case, the DNA was radioactive. All right, pretty smart, amazing experiment. Then we come across Chargaff, and he's the one that determines that all species have different amounts of A, T, C, and G, but that in every species, the amount of A and T is always the same, and the amount of C and G is always the same. Guaranteed that will be a test question for you, where you'll have to figure out how much T a particular organism, how much thymine a particular organism has, and you'll have to use Chargaff's rule to figure that out. All right, and then the last section, I'm going to skip these. Sometimes these videos are kind of bad. This last section is about Watson and Crick and Franklin and Wilkins. These are the individuals who finally discovered the double helix shape of the molecule. Watson and Crick Watson, Crick, and Wilkins, get the Nobel Prize. Franklin had passed away by that time. She died of ovarian cancer at a very young age. Is it because she was working with X-ray technologies? It was called X-ray crystallography, and she really helped them solve the double helix shape of the DNA molecule, but she did not receive the Nobel Prize, unfortunately. All right, and then we're going to move on. I think that's it, everybody. Was there anything else I needed? Oh, yeah, and then this is just sort of background information, which we already talked about. No problem with reviewing, I guess. So here's the structure of a DNA helix. When you see these ribbons, do you see this blue ribbon? That's a way of making it easier to draw. They're showing you the phosphate sugar, phosphate sugar, phosphate sugar, not having to draw it all out. So that's the phosphate sugar backbone. And when I say sugar, if it's DNA, then that's deoxyribose. If it's RNA, which does not have a double helix, then that backbone would be ribose. And then remember that these are really strong covalent bonds in the backbone. They're called phosphodiester bonds, but they're really weak bonds, hydrogen bonds, that can be broken with heat or easily pulled apart. And that's between the A's and T's and C's and G's. So between the nitrogen bases is those really weak bonds. Oh, and I will say one more thing. Don't forget about just reminding you about oh my gosh, I'm doing all the wrong things. Remind you about the 5' end and the 3' end. Remember that the 5' end, if it's at the top on one side, it's at the bottom on the other side because they're running in opposite directions, and we call that anti-parallel. All right, trying to see if there's anything else we haven't mentioned. Notice that the purines and the pyrimidines are opposite from each other. So if it has 2 rings, it goes across from a nitrogen base that has 1 ring, so that the distance stays consistent all the way down all the rungs of the DNA molecule. And then I guess the last thing that they talk about is just something from your notes here about how the chromosome is actually all twisted up. So they're looking at the different distances. So the diameter, 2 nanometers for just the DNA double helix. It starts to wrap around nucleosomes. They look kind of like beads on a string is the way they say it. So around these histones, the histones are little proteins, and the DNA wraps around them, and then it wraps around itself some more, and then it makes these big loops, and eventually a bunch of loops come together, and you end up with a bivalent chromosome that's about 1,400 nanometers as compared to each individual DNA strand, which is only about 2 nanometers. Okay, cuties, that's the background information on DNA. I hope you have a good rest of your evening.